Hello, good morning, friends. Myself, Dr. Yahya Khan, Assistant Professor, Department of Botany, J.A.T Art Science Commerce College for Women, Malegaon, is back with the another aspect, that is discovery of plant growth regulators. In the previous lecture, we had studied the plant growth regulator, introduction to plant growth regulators. Now, in this lecture, we are going to study discovery of plant growth regulators in reference to auxin. Auxin is the first hormone that is discovered as a part of the growth, or means auxin. The meaning of auxin itself, it is to grow. So for that reason, we are going to study actual in which way the discovery took place, or by which way the identification of the plant growth regulators took place. Let's see the discovery of auxin. First of all, the auxin were the first hormone discovered by the Charles Darwin, was among the first scientists to dabble or to identify or to see or to study the plant hormone research. In his book, The Power of Movement in Plants, it was presented in 1880. So the Darwin has proposed the plant hormone, was the first in his book The Power of Movement in Plants in 1980. He first described the effect of light on movement of canary grass, means the plant that he had studied, it was canary grass, that is Phalaris canariensis. In the coleoptiles, means in the stem, the upper portion, it is called coleoptiles. So the coleoptile, it is moving towards the sunlight or the effect of light on that coleoptile movement, means in that particular coleoptile there is a presence of some components and that have power and a movement in the plants, that have power of movement in the plants. That's why the Darwin has proposed this concept in the Phalaris canariensis coleoptiles. So what was the Darwin's experiment? Darwin's experiment was that with his son Francis performed experiments on coleoptile. The sheaths enclosing the young leaves in the germinating grass seedling. So the Phalaris canariensis, it is the canary grass. So in that grass he had found, or that particular plant he had taken for his study, Darwin and his son Francis. They observed that the growth of the coleoptiles of canary grass towards the light source, means the growth of coleoptiles, it is going towards the light. So that is the main aim behind his study, कि exactly which component, which molecule is been responsible for the movement of that canary grass towards the light. Followed by a series of experiments, they concluded that the presence of a transmittable substance, the presence of transmittable substance that influences the growth of canary grass towards the light, means some transmittable, means the components that are being transferred from apex to lower region, so that is responsible to give the curvature of the coleoptile towards the light. So that is nothing but it is called the transmittable substance. So that have capability, the curvature, the capability of that particular components, that particular components have the influence the growth of the canary, that have influence of the growth towards the light. This transmittable substance that now we are knowing as auxin, which was isolated by vent. Now we are knowing, but at that time, at the time of Darwin's time, there is only they concluded that what they are concluding, some transmissible substance that influence the growth of canary grass towards the light. Later it was identified, but before they were identified, they were concluded their experiment which component is responsible that particular coleoptile is moving towards the light or the curvature towards the light. Later it was identified by as auxin by Wendt. See here, the second experiment in the discovery of auxin was that particular experiment was proposed by Peter Boyson and Jensen experiment. It was called as a Boyson-Jensen experiment. In 1913, demonstrated that the signal was not transfixed but the mobile. Means what they had proposed means the signal from the apex cells to the below cells, they have signal, and that signal is due to the molecules, and that molecule or that component is nothing but it is a transfix, means it is not been present in a one cell, it is not been present at a fixed position, it is mobile phase, means some components are moving from apex region to lower region for the movement. So at that time, that is the aim of that particular or the then what they had done, they have separated the tip from the oat. Now in case of Peter Boyson, they had taken another plant, that is oat, Avena sativa coleoptile, by a cube of gelatin which prevented cellular contact but allowed chemicals to pass through. See in the diagram, you can see the gelatinous substance, it has been put into the coleoptile, means they are capable to allow the chemical constituent below, but that does not the transfer of the cellular contact. There is no cellular contact, but the chemicals are only passing through that. Only that is the concept. The seedling responded normally towards the light after taking that type of experiment. The gelatinous means in the tips, see this is the coleoptile, and in this coleoptile, that particular in between the cells of apex and base, there is gelatinous component has been put, and that gelatinous substance have no cellular contact but only the passing through the chemicals in that, and these was an impermeable substance, means there was no curvature. When the substance, the component, it is impermeable, there will be no curvature, and that particular gelatinous substance that have capacity or they are passing through the chemicals, so at that time there is a curvature. So he concluded that some substances had diffused from the tip. See, some substances diffused, there is a diffusion from tip to the through the gelatinous cut where it causes the growth. So that is nothing but they are concluded that components are moving from the apex to base. So that is responsible. Only they are telling that. In the next slide we will see. Now we will see the biosynthesis of auxin, by which way the auxin is synthesized, in the plant by biological way, that is called biosynthesis of auxin. And in this biosynthesis of auxin, that particular the representative or the indole-acetic acid, we are saying that the most significant auxin, it is the most important natural auxin, that is called significant auxin in the plant. It is mainly synthesized from the amino acid tryptophan. So, in the synthesis of auxin, there is a major role of amino acid tryptophan. So, amino acid tryptophan is a precursor for indole acetic acid, and it has a close similarity with the indole acetic acid and presumably present in all the cells. Means, in case of tryptophan, it is a close similarity with the indole acetic acid, and it is present, that particular tryptophan amino acid, it is present in all type of the cells. The progress in the auxin biosynthesis also lays a foundation for understanding polar auxin transport. Means there is a foundation put forth by the tryptophan. The auxin transport, polar transport, means from upper port to lower, that is called the basipetal succession, for dissecting auxin signaling mechanism during the plant growth or plant development. In case of when a particular component has been identified, or when we are going to study the biological synthesis of that particular components, at that time we have to study what is the signaling mechanism during the plant development. So these signaling mechanisms between the cells are been responsible in the biosynthesis of that particular auxin. C. The biosynthesis, indole acetic acid biosynthesis. That particular pathway is known as indole 3 pyruvic acid pathway. In this pathway, first of all, amino acid tryptophan donates its amino group to another alpha keto acid by transamination reaction. There is a reaction called transamination, means the transfer of amino group from tryptophan to alpha keto acid, that is called transamination reaction. There is a transfer of amino group to become indole pyruvic acid. Means tryptophan, what tryptophan does? Tryptophan does, he donates his amino group, his amine group, to the alpha keto acid component by the process called transamination. This reaction is catalyzed by enzyme tryptophan transaminases, indole tryptophan transaminases, and the indole pyruvic acid undergo decarboxylation. There is a decarboxylation, means there is a D, means the carboxyl group has been removed in the presence of enzyme indole pyruvate decarboxylase to become indole acetaldehyde. Means that particular indole pyruvic acid, then after, firstly transamination, then second is the decarboxylation. In the decarboxylation, the indole pyruvate decarboxylase enzyme responsible catalyzed by the reaction and to become indole acetaldehyde. Indole pyruvic acid is converted into indole acetaldehyde. Now see, now the indole pyruvic acid. In this pathway, first of all, the amino acid, that is the transfer indole pyruvic acid, has been formed by the enzyme, the transaminases. Finally, indole acetaldehyde oxidizes. In the previous slide we had seen these two